Our study is devoted to the use of microorganisms as potential agents for the biocontrol of sorghum disease. Sorghum is a promising crop for Ukraine primarily due to its high drought tolerance and ability to maintain stable yields under conditions of rising temperatures and water scarcity. This is especially important for southern regions where climate change complicates the cultivation of traditional cereal crops. In addition, sorghum has significant economic value as a versatile crop for food, feed, and energy purposes. It can be used to produce grain, biomass, bioethanol, and biogas, making it a valuable resource for strengthening the agriculture and energy resilience of Ukraine. At the same time, sorghum disease are one of main factors limiting its productivity and yield quality. And the most dangerous include grain mold, anthracnose, downy mildew, ergot, smooth disease, and stalk rots, which affect leaves, panicles, grain, roots, and stems. Biological control methods of sorghum are based on the use of natural antagonists of pathogenic and represent an important component of integrated plants protection. The main approaches include the application of rhizosphere bacteria, fungi, as well as microbial consortia that suppress pathogen development through antagonism, competition, and the induction of systemic resistance in plants. In a global practice, a wide range of biopreparations based on the microorganisms. The most common products based on Bacillus subtilis and Pseudomonas, which dominate the biopesticide market and have proven effective against soil-borne and fowler disease. The aim of this study was to determine the antagonistic activity against strength of Pseudomonas syringae, the causative agents of sorghum disease. The study also included an analysis of the scientific publication devoted to sorghum disease, modern methods of the biocontrol, mechanism of action, and antagonistic microorganism, as well as the particular application of biopreparations based on bacteria and fungi in plant protection systems. The study used strain Pseudomonas synxantha UCM B-399 obtained from the Ukrainian collection of microorganisms of the Zabolotny Institute of Microbiology and Virology of the National Academy of Sciences of Ukraine. Antagonistic activity against phytopathogens bacteria was determined using the perpendicular streaks method on petri dishes with PGA medium. Control plates were inoculated only with the test strain without introducing the antagonist strain in the center. The results were evaluated based on the size of growth inhibition zone of the test strains. To collect and process information, open access scientific sources were used, including databases such as ResearchGate and Science Direct. According to the results of previously conducted studies, it was established that the plant sorghum or sodium in Ukraine is affected by bacteria spot caused by strain of Pseudomonas syringae. The causative agent of bacterial spot is one of the most widespread phytopathogens responsible for bacterial disease in various agricultural crops. The percentage of infected sorghum plants ranges from 2 to 20% depending on the year and the cultivation site. The result of the antagonistic activity of Pseudomonas synxantha UCM B-399 against the causative strains of bacterial spot and presented in figure 1. The study of the antagonistic activity of Pseudomonas synxantha UCM B-399 against strains causing bacterial spot of sorghum or sodium demonstrated a high-level activity of this species. Growth on inhibition zone of 20 to 30 millimeters against the majority of phytopathogenic strains. The activity inhibited by Pseudomonas synxantha UCM B-399 against sorghum pathogens of the species Pseudomonas syringae, in our opinion, may be associated with the synthesis of biological activity compounds, particularly phenazine 1-carboxylic acid. In addition to our own experimental studies, we analyzed literature sources devoted to biocontrol of sorghum pathogens. The efficiencies of Bacillus subtilis SRB28 in sorghum biocontrol is determined by its ability to actively colonize the rhizosphere and interact antagonistically with phytopathogens. Bacillus subtilis strain used as biocontrol engines produce several key groups of biological activity substances that ensure antagonistic action against phytopathogens. The most important are lipopeptides antibiotics, including surfactant, iturin, and fungicin, which directly affect pathogen cell membranes. These compounds are present in the culture of fluid of Bacillus subtilis and are the main factors of its biocontrol activity. Their mechanism of action involves disruption of fungal and bacterial cells membrane integrity. Lipopeptides interact with the phospholipid bilayer through hydrophobic and electrostatic interactions, integrate into it and cause destabilization, leading to pore formation and loss of ionic balance in the cell. Isolated strains of Trichoderma provided inhibitions of rhizobium nihamae, growth at level of 39 to 73%, indicating a high level degree of antagonism. This effect explained by a combination of several mechanisms: rapid growth and substrate colonization, microparasitism, and production of antifungal metabolites. The mechanism of action of Trichoderma also include the indication of systematic resistance in plants, accompanied by activation of defense responses and the synthesis of secondary metabolites. Moreover, enzymes produced by these fungi are capable of degrading fungi cell walls, leading to their destruction and death, along with micellar overgrowth and the production of antifungal metabolites. The most pronounced effect on sorghum productivity is observed when Azospirillum brasiliensis is used in combination with photophosphate solubilizing bacteria. Combined inoculation significantly increase yields and mineral nutrient parameters. The mechanism of action of this group of microorganisms is associated with associative nitrogen fixation, phosphorus mobilization in the soil, and stimulation of root system development, which enhance nutrient uptake and consequently plant productivity. The use of bacteria and fungi such as Bacillus subtilis, Trichoderma, and Azospirillum brasiliensis provides combined effect on plants, combining suppressions of phytopathogens which enhance crops resistance to abiotic stress. A high level of biological activity of Pseudomonas synxantha against the majority of phytopathogenic strains was observed. The diameters of growth inhibition zone of pathogens ranged from 20 to 30 millimeters. An important feature of biocontrol agents is their multifunctionality. Antagonist microorganisms represent a promising basis for the development of environmentally friendly next generation biopopulations. Their use makes it possible to reduce the depends on chemical pesticides, decrease anthropogenic pressure on agrosystems, and ensure stable agriculture production. Reference we used, and thank you for your attention.